So if you don't know your blood types, first of all, your doctor probably knows them. So if you've ever had blood taken, then they can tell you your blood type. Second of all, if you take forensics, I know I buy stuff for blood typing there, and I think also in anatomy they do blood typing. You just have to poke your finger, and then you put the blood, dab it on a little card, and it'll show up what's positive and what's negative there. So it's pretty interesting. Now, we talk about that A, B, and O. If you are O negative, you can give your blood to everybody. Everybody wants your blood. You are the universal donor. If you are AB positive, it means, like I am, I can take all your blood. I am the universal receiver. If I'm low on blood, I can take blood from anybody. Okay? So we have the universal donor and the universal receiver. If you're O negative, who's O negative? If you're O negative, you can only take O negative blood because your body doesn't have the A protein, so it can't take anything with A. Your body doesn't have the B protein, so it can't take anything with B. And it doesn't have the Rh positive thing, so it can't take anything with that. So the only blood you can take is O negative. So you can donate it to everybody, but you can't take it from anybody. So again, it gets pretty interesting when you do that. Okay, so set up your page. Again, you guys online can pause this whenever you need to.
Can we donate to everyone because we don't have those proteins that will plug everyone?
Exactly. Now, again, you can't, it's more complex because there are a lot more proteins on that. So when we talk about donating blood or transfusing blood into a person, there's a limit to how much you can do because ultimately your body will reject it. So your body has to be making its own. Otherwise, there are going to be some problems. Sometimes it just takes a little while to make it. Now, added to that, even though I have blood that's fairly rare, my sister's AB positive also, and when she goes to the doctor to donate blood, they ask her not to because they want her to be on call. So if they have someone come in with that blood type, then they can say, okay, we can call you and you'll donate blood for us. I can't donate blood because I had hepatitis when I was a kid, so they won't take it. So if you've had diseases, certain diseases, that's why they have to fill out a form when you're donating blood. Then they'll tell you, no, we don't want your blood because you're tainted somehow. So it gets a little bit more complex than what you think. So we're doing, let's see, what is today? Is this Thursday? 4/20. See, I can't keep wanting to say it's Friday, and that's just a sad day. So 429, we're in Unit 12, Genetics. And this is period 7. So we're going to continue with B6F. And B6F says that you're going to predict possible outcomes of genetic combinations. And I'm going to put ... because if you remember, there was a little bit more to this. Predict possible outcomes of genetic combinations such as monohybrid crosses, dihybrid crosses, and non-Mendelian inheritance. The key with this, what we're going to look at today, is terms. You need some serious vocabulary to go along with this. So we need to focus on some terms that you're going to have to know because when we describe things, when you get asked genetics questions, they're going to throw these terms around like crazy. So you have to know these terms. So we're going to start off, and I have 13 of them. I'm not going to give you some massively detailed definition out of the book. I'm going to keep it really simple and straightforward, something that I hope you can kind of keep in mind when you do this. So let's start off with number 1. Number 1 is genotype. So genotype would be the actual genes in an organism. Its genetic makeup. Another way of saying that is talking about its genetic makeup. When I ask you for the genotype, I expect you to give me, we usually symbolize them with 2 letters. So when we talk about eye color, you can be dominant, pure dominant. So I want the genotype, you're going to give me the 2 letters, big B, big B. Or maybe you have, you're heterozygous for that, or hybrid for that, so you have big B, little b, right? So when I ask you for the genotype, I want the 2 letters. If you just say BB, that's not enough. You got to be specific. We say dominant traits are capital B, lowercase traits are the recessive traits, right? So you have to be very specific about that. Going hand in hand with that is another word called phenotype. So number 2 is phenotype. These two are huge. If you don't study these two words, you're going to get thrown off. You're going to miss a lot of questions from this. Phenotype is the expression of the gene. Okay, so I'm going to say this in 2 ways. It's the expression. It's what the organism looks like. Okay, so I don't want letters. When I ask you about the phenotype, no genes, no letters. If you try and say the letters, I'm going to say, nope, that's wrong. No letters, genes. I want you to tell me if capital B is brown and lowercase b is blue, what's this person look like? Brown. We'll say brown eyes, right? What does this person look like? Brown eyes. That's what the phenotype is. It's just what do they look, how are they showing it? Now with blood type, I can't look at you and tell your blood type, right? So we'd have to get some tools out to determine the blood type, but that's still a phenotype. What is your blood type? Well, my phenotype for blood type is AB positive. Yeah? So that's what phenotype is. Never want to know the letters. I just want to know what does it look like. Okay? And you understand that human genetics isn't the same as plant genetics, and that's not the same as dog genetics or anything like that. We can't take our characteristics, dominant recessive traits, and how they show up in humans and apply them to dogs or apply them to corn or something like that. So we look at the species and look at the genetics there, and that's how we determine what's going on with that. Okay, number 3 is gene. And when we talk about a gene, this will be the 5th time I think you've written this down. It's a section of DNA that has the information to make one protein. It's a piece or a section of DNA. And DNA, if it's all wadded up in nice little chunks so we can see it through the microscope, we call those chromosomes, right? So a gene is a section of a DNA or chromosome that has the information to make one protein. And remember, when we're talking about making proteins, we're talking about our traits, our characteristics. They go hand in hand together. That's what we're dealing with here. Number 4, allele. And I'm going to put it as plural. Alleles. Alleles are genes that are coding for the same trait. Remember we talked about your genes coming in pairs? We call those alleles. Okay? So genes coding for the same protein. And remember, I'm going to put in parentheses, we're talking about traits. Maybe that protein's your eye color protein. Maybe it's your skin color protein, your hair color protein. Maybe it's whether or not you have the protein that enables you to roll your tongue. All of that's a part of that. So these are genes that are coding for the same trait, and they are found at the exact same position on homologous chromosomes. Homologous chromosomes. Remember homologous chromosomes? You remember you have your chromosomes come in pairs, right? So you have 2 of the number 1s. So these are the chromosome, and we can say it's mate, like a sock has a mate. So if I look at make it a good one. So if we did, where's that? Oh, here we go. So here's your mom, here's the sperm, here's the egg. So we got, this is the number 1 chromosome, and this is the number 1 chromosome. If we're talking about alleles, it would be a gene that's right there, and its mate over here. It's the exact same position on the chromosome. It's just this is the first number 1, and this is the second number 1 chromosome, right? So if they're alleles, they're going to be coding for the exact same thing. They're going to be in the same position on those matching chromosomes, right? So that's what alleles are. A lot of times when we refer to genes, we're talking just in general. Here's a gene over here and here's a gene over here. Alleles are always talking about the fact that, hey, we're talking about these two things together here. Next one is homozygous. Okay, that is a big, ugly word here. First thing I want to point out is this zygous part. Whenever the sperm fertilizes the egg, immediately it's called a zygote. So the second after that sperm fertilizes the egg, the whole thing, the egg that's been fertilized, has the whole package of chromosomes. We don't call it an egg or a sperm anymore, we call it a zygote. So that's where they're coming along with this word zygous. What's the prefix homo mean? Same. So in this case, if it's homozygous, it's the same, or we call it pure, right? Pure, it means the alleles are identical. That would mean they would both be big B, or little b, little b. They're pure for that trait.
That's how my blood type is.
Yes. Yeah, it'd be O and O. You'd be homozygous for that trait of blood type. Would I be homozygous for my blood type? No. So what would I be instead? It's not homozygous. What's the opposite? Heterozygous. Now, heterozygous, that prefix hetero means what? Different. And we call this also hybrid, which I think you guys knew those words the other day when we were talking about this. This means the alleles are different. So in this case, you'd have big B, little b. They're both still talking about, eye color, or they're both still talking about rolling your tongue. It's just that one will be dominant, one will be recessive. Okay, number 7, and I think some of these terms you won't have to study. I think you know them, but sometimes it's the way they're described, which on the test can kind of throw people off. They're supposed to be, by the way, giving me the results for the test sometime this afternoon. It's not in there yet, so probably tomorrow we might have a chance to look at those. Okay, so number 7 here, dominant allele. Okay, the dominant allele is the allele that is expressed in a hybrid. Here's the word, again, expressed. That's the one, this is the one that the protein's actually made. Because you have 2 genes for the trait, typically in Mendelian genetics, only one of those proteins is going to be made. So, in most cases, in normal Mendelian genetics, then let's say we have brown eyes or green eyes, right? Only one of those is going to be expressed. You're not going to have one brown eye and one green eye, right? If it's a mutation. Usually if there's a mutation, it'll be one color of eye and then blue, where it's actually the blue in that case is usually a lack of color. So for humans, a lack of color means it's going to be blue. In animals, a lot of animals, when they're albinos, they have that lack of color and it's kind of pink. It never shows up like that in humans. Yep. At least not in live ones. So we have the dominant allele. When it says expressed, this is the one that's made, like the protein that's made or shows, the one that shows up. So if we're talking blood type, then it might be the one. Okay, so that's a bad example since that it's talking about some protein inside the blood that's floating around inside there. If that's present, that's the one that shows up. It doesn't mean it's always easy to see, right? Always keep that in mind. There are a lot more traits that we can't see, but we can test for, rather than just the easy ones that we can look at and say, oh yeah, I see that. Okay, number 8, chromosome. Oh, sorry, wrong one. Recessive allele. This is the allele that's not expressed in a hybrid. It's there, but you never make that protein. So that trait is not shown. It doesn't show up like that. So when we talk about expressed, we're talking about making the protein that the instructions from the DNA came from, the gene has. And typically we look for those proteins that show up. Yes? We want to see the ones that are obvious. You know, most people generally are. There are certain traits we can look at and we can say, yeah, okay, that's pure or homozygous. But most of them, I mean, we have such a broad range of humans and we kind of spread our genes all out over the place that we tend to be a lot more hybrid. We're mutts. Yeah.
That's why we're lasting.
Yeah. Okay, number 9 is chromosome. And remember, what chemical are chromosomes made of? You've heard of that stuff before, DNA? What chemical are chromosomes made of? The whole thing, what's it called? It's called DNA, right? Now we can talk about what DNA is made of individually, but when we talk about chromosomes, well, it's just a big, huge chunk of DNA, right? It's got some proteins on it because the chromosome, you take the big, long thing of DNA right here, and you package it up. And that's what we call a chromosome. It packages up, it's got like twine or something. You know how you have something long and you have to wrap it up and it's got to be kind of tied up, right? It's got this twine, if you want to call it that, of proteins that hold it in a package here. So a chromosome is a coiled up strand of DNA. And I'm going to put in parentheses, and I'm going to tell you it's contained by proteins. And when I say contained, it means it's holding it in. It's like handcuffs or something. We can only see these when the cell is going through mitosis, when it's reproducing. The cell's going through mitosis, making new cells. That's the only time we can see chromosomes. Each chromosome has thousands of genes on it. What's another word for the genes that we talk about? Starts with an A. Alleles. Or allele. You'll hear it pronounced both ways. Number 10. Karyotype. Okay, so the karyotype, look on page 3 of your packet. Somebody tell me what a karyotype is. Page 3 has some karyotypes. What are those? Anybody, what are those things? What's it look like? What are those things? They're chromosomes. Is this chromosomes from is this one person? Okay, so This is a picture of the complete set of chromosomes for one person. That's what a karyotype is. Yeah? So a karyotype is a picture of a complete set of chromosomes. There we go. And then the next one, number 11, I have 13 of these, so I'm going to do number 11 here. Pedigree. You guys, I think you've heard of that word before, yes? Pedigree. A lot of times if you talk about dog breeding or horse breeding, or if you guys are in ag, you talk about hog breeding, things like that, and you look at the pedigree, if you've got race horses that are really famous, they won a lot of races, they look at the pedigree because they want those genes to continue on, right? So when they look at a pedigree, they kind of look at their family tree, their history. Usually though, when we talk about pedigree in biology, we're really focused on specific traits. Not the whole thing in general, but really specific traits. So a pedigree is a diagram, and you guys have probably seen these, I don't know if you've paid attention to what they are. It's a diagram of family inheritance of traits. And it's by generation. So when I say by generation, let me see if I have a picture in here really quickly that I can find. Oh, so they look a lot of cases like this. Have you seen charts like that? Yeah, that's what it is. Usually circles are female, males are square. Yeah, that's generally the way it is. But they usually just focus on one particular trait at a time. So a lot of times if they color it in, that means they have that trait. If they leave it blank, then it means they didn't have that trait. And so they can actually even say, oh, well, if it's half colored in, they think there maybe were hybrids. They can tell if they're hybrids in that case. Now, I'm going to skip a line, and what I'm going to do is I need you to skip this too, because the last two are all about non-Mendelian genetics. So skip a line and put little stars beside these. But number 12 is codominance. And codominants are when both traits are expressed. There isn't one that's dominant over the other. They both show up. Not blended, they're just both expressed. So think blood type. Blood type is a really primo example of this. Again, you can't apply, you have to look at the genes for different species. You can't apply them all at the same time. But so here is Z. He is showing codominance. So you see both the black and the white showing up in him. Yeah? With Pixie over here, she has a tiny little bit of tuft right here, but basically she's, by far, most of her skin is showing the black dominance, right? So in particular dogs like this, when they have them, they have codominance. Both of these colors are showing up. If you look at the skin, if you shaved them down, their skin would be spotted like that too. Yeah. The second one on here, number 13, last one that we're going to do. Number 13 is incomplete dominance. And it's different from codominance. Incomplete dominance. And incomplete dominance is when one doesn't show up, the other doesn't show up. They both show up, but they blend. So incomplete dominance, they have blended traits. A lot of times it's easiest. There are certain classic examples of this. Do you guys know what snapdragons are? Flowers? Snapdragons? No? Yes? You've heard of those? They're cool little flowers. I tend to kill stuff, but I was able to grow some one summer. And they're they have like a slew, like they have a stalk and they have lots of little flowers on there. And if you take the flower off, like pull the flower off, and you pinch the back of the flower, it opens and closes the mouth like a little dragon. That's why it's got its name that way. If you have a red snapdragon flower and you made it to a white snapdragon flower, you don't get red, you don't get white, you get pink. Yeah? So that's what I mean by blended traits. You don't get one they just blend smoothly. You're not going to have splotchy red and white. That would be codominant. So if you had spots of red and white, that would be codominance. When you blend the two colors into pink, that's incomplete dominance. Mendel didn't know about this, so over here on the side, I want you to put non-Mendelian. This is your non-Mendelian genetics. Non-Mendelian. He didn't know about this. Non-Mendelian inheritance. Okay? So these are the terms you're going to have to know. If you guys don't spend some time on these terms, you're going to flunk the test that you have. Okay? You need to make sure you understand the terms. Some of them you already know. Yes? Some of them you just need to get familiar with the way you might know what it means in general, but when you say the words, like when I give you the scientific definition, it's not quite clicking with you. So you need to study that there too. What I'd like you to do is flip over to page 13 in your packet. Once you have that written down, page 13 in your packet. Okay, so when we were looking at this Tuesday, we talked about, we have two tall hybrid pea plants, we crossed them. Then we had a pure tall pea plant crossed to a pure short pea plant. And we had questions. This last one is an example of non-Mendelian. I'm going to put non-Mendelian, okay? So when I look at this, I see two pink snapdragons are crossed. Fill in the Punnett square below to show all the possible combinations of sperm and egg. Notice they gave us a key. R is red, capital R is red, capital W is white, and if you have the R and the W together, you get pink. So look at this. I have a sex cell with an R, a sex cell with a W from one organism. I have a sex cell with a capital R and a capital W from the other organism. So fill in your Punnett square if you haven't already. When you fill in the Punnett square again, both letters, this letter drops down into both boxes below it. And then on the side, the letters slide in to both boxes beside it. Okay? How many different phenotypes could appear in the offspring? 3. What are they? Yeah, those are your options, right? And I see, what color is this one? Red. What color are these two? Pink. Even though I put the W in front, that's still W and R together, right? And what color is this? So I have three colors right here, yes? What fraction of the possible offspring have the same phenotype as the parent? What color are the parents? What do they look like? Okay, 2 pink snapdragons, R and W. It's not together. This is incomplete dominance. Incomplete, which means they blend. So the parents are pink. What fraction are also pink in here? 2/4. I'm going to keep the 4 there because we're going to go and add these numbers up in a minute. And then what fraction of the possible offspring are red? Which one is it? Big R, big R. So 1 out of 4. What I want you to do now is I want you to add all these numbers up right here.